Hello, I am Javier González Canales, Research Assistant in the Department of Edafology at the Politécnica University of Madrid, and I'm going to talk about the poster called "Can Irrigation Modulate Soil Microbiological Response to High Levels of Tropospheric Ozone?" High tropospheric ozone concentration show adverse effects in agrosystem. Ozone is a potent oxidant that causes damage to plants and can reduce the carbon transfer to plant roots with change in root composition and exudates, thereby affecting the abundance, metabolism, and composition of soil microorganisms as well as nutrient cycling. These effects depend on factors such as plant variety, ozone doses, or soil type. Water deficit seems to modulate the ozone effects on plants and presumably on soil microbiota. Although soil moisture is an essential factor in explaining soil microbiota structure under high ozone conditions, there are few studies that have investigated how the effects of irrigation is transferred to soil microbiota. For this purpose, a field trial was carried out on experimental farm La Higueruela in Santa Olalla, Toledo, in the center of Spain. The trial was conducted with open top chambers, OTCs, in a split plot randomized block design, with 3 replicates. The main factor was 4 ozone levels: filtered air, non-filtered air, ambient plus 20 ozone ppbs, and ambient plus 40 ozone ppbs, which were randomly distributed in the OTCs. In each half of the chamber, one of the irrigation levels was randomly applied. The ozone-sensitive spring wheat variety Califa Sur was chosen as the crop. The plants were exposed to the different ozone doses and irrigation until their maturity. Soil sampling was carried out in May 2020, when the wheat was in the grain filling stage. In each treatment, 2 plants were randomly selected, and after separating the shoot root with their soil was extracted with a cylindrical sampler. The samples were divided into portions 0.5 cm deep and 5 to 20 cm deep, and kept out in cool until the laboratory. Each sample was carefully processed to separate the roots, the soil reservoir 2 mm around the roots, and the rest of the bulk soil. Soil samples from the 2 extractions were mixed to give 1 sample per treatment and were prepared and stored until further analysis. In each fraction, reservoirs and bulk, several chemical and microbiological properties of soil were measured: pH and electrical conductivity, total carbon and nitrogen, melich extractable elements, nitrate and ammonium, basal and substrate-induced respiration, microbiological biomass carbon and nitrogen, dissolved carbon and nitrogen, soil DNA extraction, quantification of bacterial, archaeal, and total fungal genes by qPCR. That analysis were carried out by ANOVA with generalized linear model for a factorial block design with split plots. We found that most of the root biomass, around 19%, was found in 0.5 cm deep, where the soil analysis were carried out. The reservoiric soil fraction showed higher sensibility than the bulk fraction to ozone level and water availability, confirming that the effect of ozone on the soil microbiota is through the effects on the wheat plant. Soil chemistry variables were much more sensitive to water availability than the ozone level. Interaction between both factors was rarely observed. Micronutrients respond to ozone to a greater extent than macronutrients, especially cobalt, manganese, copper, and iron. Microbiological and molecular variables were more sensitive to ozone than chemical variables. In some cases, the irrigation level modulated their response to ozone levels. Non-filtered air was the less sensitive treatment to irrigation levels, whereas non-filtered air plus 20 was the most sensitive. microbial biomass, carbon and nitrogen, showed significant declines in the ozone-enriched treatments, regardless of the irrigation level. The abundance of total bacteria and total fungi increased in the ozone-enriched treatments, especially when the irrigation level was optimal. As conclusion, the microbiological response of soil to tropospheric ozone pollution in a Mediterranean wheat agrosystem is complex and can be modulated through irrigation.